Hello, everyone. My name is Noah Weisleader, and I'd like to welcome you to the GNEM Symposium Speaker Series presented by the Neuromuscular Disease Foundation. I'll be administering today's call. My name is Noah Weisleader. I'm a professor and director of graduate studies in physiology and cell biology at The Ohio State University. And it's a great pleasure to be here today, again with you in this series to talk about GNE myopathy and some of the efforts involved in the study and treatment of the disease. I'd like to give a quick reminder that if you have any questions during today's seminar, we're using the online question and answer feature of Zoom. You should be able to access the Q&A tab towards the bottom of your screen, depending on how things are configured. But if you have questions, please enter them into that section, and we can, if they're urgent, let me know. Otherwise, we will probably hold these till the end. And then during the at the end of the session, I will administer the Q&A portion using those questions, as well as probably some other ones that come up. I would also point out that we have a couple of other events coming up in this seminar series in the near future. I wanted to point out that Dr. Paul Martin will be presenting on December 9th, and then that Dr. Francis Rosignol will be presenting as well on December 18th. So we're excited to have those two coming up very soon, and there'll of course be announcements that go out about that as well. But today we're really excited to have one of our rising stars in the GNE myopathy field here to give our today's presentation. Today it will be Katherine Koczwara. Katherine Koczwara. She's currently a PhD student in the LEC lab at the Yale School of Medicine. She received her Bachelor's of Science in Biological Sciences from the University of Notre Dame, and then went to work at Yale on developing her PhD. Her studies focus on concentrating on both computational biology and genomics, and combining those two interests in order to study human genetic diseases, and with a particular emphasis here, as you can see, on GNE myopathy. She's been investigating the application of CRISPR-Cas9 technology in terms of editing to correct patient mutations in GNE myopathy, as an example, and also has been instrumental in establishing a biobank of GNE patient cells, many obtained through donors by the NDF. And this has become a really cornerstone resource to be able to spread the amount of GNE research that's done, both in the United States and throughout the world. So we very much appreciate your research and the effort you put into helping to build the field. And I will turn this over to you today to present your talk on developing an AAV gene therapy for GNEM.
Hi, thanks, Noah, for a great introduction. So I'm going to be talking a bit today about the work that I've been doing in the LEC lab regarding to developing an AAV gene therapy for GNE myopathy. Dr. Julie Crudell gave a part 1 to some of this work at a previous seminar, so I highly encourage people to watch that as well. But diving right into kind of the work that I've been doing on this project is the aim of the project as a whole is to develop an AAV gene replacement therapy for GNE myopathy. Kind of the work that I've been doing has really been focusing on optimizing a part of the AAV called the transgene cassette in vitro, so in cell lines, before moving to more in vivo mouse studies. And so first, a bit of background into GNE myopathy. So GNE myopathy is, as many people know, an autosomal recessive disorder that leads to progressive skeletal muscle atrophy and weakness in the legs and arms. It's caused by mutations in a gene called GNE, affects about 1 in 1 million people worldwide, and characteristic of the disease are characteristic rim vacuoles and inclusion bodies, which you can see here, some of the staining in the GNE myopathy patient. You can see these inclusion bodies and rim vacuoles that you don't see in healthy muscle tissue. So as I mentioned, GNE is caused by mutations in a gene called GNE, which is diagrammed here. So there are 2 domains of the gene, an epimerase domain and a kinase domain. As you can see, with some common mutations listed across the bottom, these mutations are spread all across the disease, and there are many different mutations that have been found to date to cause GNE myopathy. And lastly, GNE is an enzyme in a sialic acid biosynthesis pathway. So it's here diagrammed is the sialic acid biosynthesis pathway, and you can see that GNE performs 2 of the steps in this pathway, the epimerase function and a kinase function. And eventually this will lead to the production of sialic acid, which is then used to sialate proteins on the cell surface. So in terms of developing a treatment for GNE myopathy, there are kind of 2 main ways to think about doing this. The first is by supplementation, something like sialic acid or Manac supplementation that some collaborators at the NIH are working on. And this aims to supplement with some of the compounds produced in the sialic acid biosynthesis pathway. So not targeting GNE, but looking to add some of the compounds that it would normally produce that it can't in patients with GNE myopathy. The second approach, and the one that I'm going to focus on, is a gene therapy or gene replacement strategy. So this aims to replace the mutant GNE and add a healthy copy of the gene, introduce that into the cells. So the healthy copy can then produce the sialic acid. And so I've mentioned a lot gene replacement therapy, but for those of you who don't know, I can go a bit into what that is and what that entails. So gene replacement therapy aims to deliver a healthy copy of the GNE gene to cells, affected cells. So the cells that don't have a mutated GNE gene. We want to introduce a new functional gene into the cell. And so this new functional gene can work in the cell function. In this case, GNE enzymatic activity can be restored. And so the way gene therapy works is AAV vectors are used to deliver healthy genes to the cells. So in this case, a gene cassette pictured here is packaged into an AAV vector, and this combined vector is introduced into the cells, which then allows the gene cassette, in this case, the GNE gene, to be expressed. And so once that gene is delivered to the cell, it undergoes a process in which the DNA that we deliver is transcribed into RNA, and this RNA is then produced into a protein. So we can kind of use this to measure how well our delivery is working by either looking at RNA or protein expression to see if we see the GNE gene expressed. So the first step in the work that I've been doing in the lab is first to design this gene cassette for GNE myopathy. And so there are a couple of key components that I wanted to optimize that would lead to the best or the highest GNE expression. The first is the promoter. So this is the thing that's driving the expression of the gene. Second is an intron that is between the promoter and the transgene. And then lastly was this transgene itself. So which kind of version of the GNE gene that we want to use. And so the way that I did this was generating a bunch of different vectors that have different combinations of promoters, introns, and transgenes, introducing them to cells in cell culture, and then harvesting the protein and looking to measure how much GNE protein expression we can see with the variety of different vectors that we have, and see which one leads to the highest GNE expression. And so the first thing that I wanted to look at was which promoter to use. So since GNE is a muscle-specific disease, we wanted to use a muscle-specific promoter. So there were 2 of interest that we wanted to use and test to see which one worked well. The first is called CK80, and the second is MHCK7. And so I created constructs that have either of these 2 vectors, introduced them into cells, and here you can see is a blot of just looking at protein expression. So we can use an antibody for GNE to look and see how much protein was in all these samples. So we're looking at the higher band here. We can see in lanes 4 through 7 are constructs with the CK80 promoter, and then lanes 8 through 11 have constructs with the MHCK7 promoter. And what I hope you can see is that in general, these lanes, especially in lanes 4 and 6 that have the CK80 promoter, have stronger bands, meaning more GNE protein was expressed using this promoter compared to the MHCK7 promoter. So that's the promoter that we decided on moving forward. And the second thing that I wanted to look at is the inclusion of an intron. So you can either have an intron here, and we're using the SV40 intron, or no intron at all. And so I created a bunch of constructs. The ones that have the SV40 intron have a red asterisk, and those that do not have a blue asterisk. And so what I hope you can see is that in all the lanes that have the red asterisk, we have a very strong band, meaning a lot of GNE protein was expressed from the constructs with the SV40 intron present, compared to the lanes with the blue asterisks that don't have as high a stronger band, meaning not as much protein is expressed. So we concluded it was very important to have this SV40 intron included in our construct. And then the last thing that we kind of wanted to look to optimize was which transgene that we wanted to use. So GNE has a few different isoforms. And what we wanted to see is, since GNE is a muscle disease, we wanted to see which of these isoforms was expressed highest in the muscle tissue. So fortunately, we had a few samples from healthy donors, healthy muscle samples, and looked to see in these healthy muscle biopsies, which RNA, the RNA from which isoform was expressed higher. So we did an RT-PCR. And what you can see is in these 4 lanes, that looking at the GNE2 isoform, they're very weak or faint, meaning not a lot of GNE2 isoform was present, compared to these 4 bands was looking at the GNE1 isoform. And they're very strong, meaning that in these muscle samples, we saw high expression of the GNE1 isoform. So that's the one that we wanted to use moving forward. So basically, in conclusion from all of that work, we're looking to kind of create this construct that has the promoter, the intron, and the transgene. Our lab came up with this construct that has the CK80 promoter, the inclusion of the SV40 intron, as well as using the GNE1 isoform, which we also tagged with the protein tags, so we would be able to detect it later. And also previous work done in the Matrani Rosenbaum lab has also developed a separate AAV gene therapy that uses this construct that has a different promoter, the MCK promoter, but then the rest of the construct is very similar with the SV40 intron and the GNE1 isoform. So we wanted to test both of these constructs from the one that we developed, as well as the work that the Matrani Rosenbaum lab has done. And so again, we introduced these into cells and looked to see if we saw GNE protein expression. So as you can see, in lanes 5 and 6 were our 2 constructs, and you can see both had very strong bands, meaning very strong GNE protein expression. In several different cell lines, both in a human cell line as well as in a mouse cell line. And we also looked at the RNA that was produced when we introduced these constructs into cells. And you can see that at the RNA level, both the CK80 construct from our lab, as well as the MCK construct from Dr. Matrani Rosenbaum's lab, also had high RNA expression. So both of these constructs show good promise in being used for an AAV gene therapy. So kind of the next step was to take these constructs that we had optimized in cells and designed in cells and move forward to a dose and biodistribution study in mice. And I mentioned, but worth pointing out again is this effort is a very collaborative effort from many labs and many groups. And I'm just working on a very small part of this step right here, but have had many collaborators that we're working with and are currently working on some of the mouse studies right now. And so the way this first dose of biodistribution study work is we take the constructs that either the Leck lab or the Matrani Rosenbaum lab have created. We generate the AAV9 particles containing our cassette of interest. We'll dose at different doses into mice, and then we can look to see, we'll harvest RNA or protein from different tissues and look to measure the GNE expression that we're getting from our AAV. And so I did this first looking at several different muscles at the RNA level. So looking at the exogenous GNE1 expression in the gastroc muscle. And you could see here, compared to mice that didn't have any construct, we see an increasing response of low dose for both the MCK construct as well as the CK80 construct. We see GNE expression at a lower dose, and then when we use a higher dose dosing these mice, we get even more GNE expression. So we see a dose-dependent GNE1 expression in the gastroc muscle. We see a very similar thing in the quadricep muscle, which is what we expect because our promoters are muscle specific, where we don't see any expression if the mice are not treated. Low expression if they're treated with a low dose in either of the constructs, and then a higher GNE expression when we use a higher dose. We also look to see in liver because we want our therapy to be relatively muscle specific. And while we did see a little bit of expression in the liver, dose dependent as in the muscle, when we normalize across tissues, you can see that in the liver, we had very, very little expression compared to the two muscle samples we looked at, like in the gastroc and the quadricep. So this is an encouraging result that we're seeing good RNA expression of GNE1 across our two muscle tissues that we've looked at so far. We also wanted to try and look at the protein expression. So again, going from RNA to protein to see if we get strong gene protein expression. So we're looking for strong bands, and we hope to see, as with the RNA results, that we wouldn't see a lot of expression in the untreated samples, a little bit of expression in the samples treated with a low dose, and then higher expression with this in the samples treated with high dose of these AAV gene therapies. Unfortunately, that didn't seem to be the case. We didn't see a strong pattern in either the gastroc muscle or in the quadricep muscle. It was not a very strong trend. So we're still working on doing a few things to see if we can find a better way to detect this GNE protein in these samples. And that's where we're currently at, is we're still working through a variety of more tissues from these mice, as well as looking to optimize being able to detect the GNE protein expression. And so following this biodistribution study, where we're looking across more tissue types and looking to optimize detecting our protein expression, we also are in the process of trying an even higher dose that we're looking for to see if that will improve our ability to detect protein expression. So that is in progress. And then also work at the NIH is currently being done to characterize and treat a mouse model of GNE myopathy. So they're working to characterize it now. So we would be able to do a future efficacy experiment. So kind of key takeaways of what I've been working on and where we hope to go is that we've optimized some AAV vectors in vitro and shown that the CK80 SV40 GNE1V5 construct results in high GNE expression in myoblasts. Some preliminary results of the dose and biodistribution study in mice show dose-dependent muscle-specific expression using both the construct we developed as well as the one that Dr. Stella Matrani-Rosenbaum developed in her lab. And sort of future and ongoing work will aim to look to just show that we're getting strong GNE protein expression, and then as well as look to move into a disease mouse model to determine the efficacy of any potential gene therapy treatments. So with that, I'd like to thank everyone in my lab, and especially the NDF for funding a lot of this work and giving me the opportunity to present today. So thank you, and I'm happy to take any questions.
Thank you very much for that update on the progress relating to both vector development as well as the biodistribution studies, which are very exciting. It's seeing them go into mice and start to understand which one of these are going to be effective in gene therapy is going to be very helpful. So we have a couple of questions coming in. Let me start with the first question. Are your studies with normal myotubes? Have you tested in GNEM myotubes as well?
Yes, so actually we have tested in both normal myotubes as well as the GNE myopathy or knockout myotubes as well. And both in our studies showed that using our constructs, we were able to get strong wild type exogenous GNE expression from our constructs in both the normal myotubes as well as myotubes that are missing the GNE gene.
Right. Maybe as a follow-up that I could throw in there too. Is that now looking at wild type mice with the biodistribution study, do you think that's one of the reasons kind of obscuring a clear conclusion there, because you have the native present there as opposed to what's taking place in the maybe with a knockout mouse or something like that?
Yeah, exactly. So we do have the endogenous GNE expression from the healthy mouse. And we're trying to detect any exogenous, so any GNE expression that we're introducing over that, which could be making it difficult. So we did add a protein tag to our exogenously expressed GNE. So we added a V5 protein tag. So I'm currently working on some things to hope to use that to be able to distinguish between the endogenous GNE of the healthy mouse compared to the exogenous that we're expressing from our construct.
That makes sense there. Additionally, I wonder if there's, just to follow up on that as well, you had some variation there in the protein level of expression, whereas the PCR was pretty consistent, or looking at the RNA level is pretty consistent. Do you see variation in the level of, it looked like you had it just even in control background too. So do you think that's consistent, that you see kind of variation in the level from one mouse to another mouse, even in the same anatomical muscle?
Yeah, hard to tell. Given that our loading, some of our controls were also pretty variable. So I'm going to repeat a couple of times and see if we can get a clear answer to that question. But inconclusive is for now.
Sure. I mean, but you do see that with some proteins. You do see variation even in a well-defined genetic model. You can see variation, time of the day, and lots of different things that can contribute to that. Anyway, another set of questions coming in here. This is actually a two-parter, so I'll start with that. First, say thank you for a very interesting talk. And once ready to use patients, would AAV gene therapy in theory be a single treatment therapy, or would multiple rounds of gene therapy be necessary?
Yeah, so typically AAV gene therapies, at least as they're currently used, tend to be one dose. But as a still emerging field, lots of things are still changing, and people are making more progress with the delivery method. So that's kind of outside the scope of what I was working on in the study, but definitely something to be considered.
Hi, I can jump in. This is Angela. Like currently, in terms of AAV gene therapy, it's a one dose deal, because once you dose, your body develops antibodies to the virus, which prevents them from transducing muscle cells when you try to do repeat dosing. But having said that, there are some emerging technologies in the field now that are looking to address the redosing issue. The idea is that right now, anyway, what gene therapy drugs aim to do is to hit all the muscle fibers all in the one go, so that we get as much expression of the missing gene, of the healthy gene, in the one shot, so that it doesn't need to be repeated. But in the event that it does need to be repeated because maybe the transgene has dropped out or has been diluted in some way, then we're going to need to rely on newer technologies to lower antibodies in individuals in order to permit redosing. I hope that answers the question.
Yeah, I think that addressed that. Actually, there was a follow-up question. I'm sure they can follow up too if they have a follow-up question. But second part of that question was somewhat related to what you were just saying, but sort of the other side of that coin. Would there be any risk of immunoreactivity that would be taken into account when designing a final therapeutic approach?
Immune reactivity, yeah. So currently in the field, there's definitely immune responses to AAV. Some people handle it well, some people don't, and that adds to the risk of delivering AAV gene therapy. And currently the field is trying to work together to try to figure out who is at risk, because so it seems like some people just don't take it well and are at risk of death, and we still, there is a lot to figure out in terms of the safety of AAV gene therapy. But currently it is the most efficient way of delivering a gene, a healthy gene into a human that we have to work with.
Yeah, I think maybe they were also one aspect of that, not just the AAV itself, but the expression of GNE in the specific context. And there, I think it would be less, they'd be less concerned about the immunogenicity of the GNE protein exogenously expressed, since there's the endogenous protein. Most patients, they're still expressing the endogenous protein usually with a small mutation in it. So that, do you think that would affect the likelihood of immune response to the actual expression of the transgene?
Yeah, I think that given that most GNE patients have a mutant form of GNE as opposed to not expressing anything, that's sort of desensitizes the immune system, and sort of when we go to introduce the healthy copy, the body would have sort of seen the protein before, as opposed to not having seen the protein at all. I think there's usually very little difference in the structure of a mutant GNE protein with one missense mutation versus like a healthy copy of the GNE gene. Sorry to barge in, but Catherine, if you have anything to add, sorry that I jumped in there.
No, it's great to hear your input.
Great. And always, of course, feel free to put any follow-up questions in the Q&A box. Another question, is the aim to recreate normal levels of GNE expression in muscle cells or to exceed that normal level?
Well, obviously normal would be ideal, but we'll see what the mouse tells us, you know, what levels. Maybe we need to outcompete the mutant GNE in order for the wild type healthy copy to play the more dominant role. So this is something we also need to figure out. But the good news is that studies by previous groups such as Stella Rosenbaum have shown that overexpressing GNE is actually does not have overtly toxic effects. So we don't really have to worry about that because of, I think, the negative feedback inhibition of the GNE gene. We're pretty safe in that we're not going to make too much sialic acid if we were to introduce a lot of the GNE gene. There are sort of molecular feedback loops that prevent the pathway from being on overdrive.
Yeah, as most metabolic pathways have multiple levels of regulation, so that is, I think, a reasonable expectation. Catherine, anything you wanted to add there?
No, that's great.
Another question, is there any consideration as to how one might ensure efficient balanced distribution of AAV to skeletal muscles in patients?
Yeah, so that I talked today about basically the transgene cassette that's packaged into the AAV. So the actual distribution to skeletal muscle is controlled by the serotype of AAV used. So not in our lab, but there are many labs out there that are working on improving and finding different and optimizing serotypes of AAV. So in this case, we used AAV9 that would address that question, but not something that we're currently working on doing.
Great. Another question, robust discussion here. That's great. Please keep the questions coming. Do you have any data on the amount of GNE protein required for a therapeutic purpose?
So that would be, yeah, so that would be kind of the next future work in hopefully a disease mouse model would be the point of that efficacy study, would be aimed to determine exactly that, how much healthy GNE expression we would need to have a dose that was effective in restoring it. Phenotype.
Sure. And when you label them as low and high doses on your figure, trying to keep it accessible to everybody's level. But I think really what you're listing there as the high dose kind of falls in a medium dose for what's used in mouse models for neuromuscular disease at sometimes, right? So in theory, you can envision going to a even higher dosing level potentially if necessary. Isn't that the case?
Yeah, so the ones that we did currently for people were 1E13 vector genomes per kilogram and 5E13 vector genomes per kilogram, which are both relatively low doses in terms of how high you can go. And so in progress, we've also dose of mice at 3E14 vector genomes per kilogram, so a very high dose, and are waiting for the mice to age. So hopefully that we'll see kind of the effect of a higher dose as well.
Right. Right, but that'll certainly give you a better idea how much you'll certainly need for that therapeutic purpose for sure, or at least set it up to do it in another mouse model where there's a phenotype to correct. Excellent. Is there any possibility that different forms of GNE have greater expression in different muscle types, I guess?
Yeah, so that's if you're talking about the different isoforms, which I see a couple of questions about. Yeah, so that's something that people have looked at. I believe Marianne's group or Stella's group have done and looked at that and looked at the different GNE isoform expression and concluded on using GNE1 in general for muscle therapies. But there are, several different isoforms that other labs have looked into to see which is expressed across different tissues.
Right, and and so you went with the GNE1 just based on that literature before, on the previous literature for that.
Yeah, both previous literature, some information on GTEx, and then also our own study looking at healthy muscle samples saw that we saw more GNE1 expression compared to GNE2 in some control samples that we had in the lab.
Great. What cell lines were the AVs tested on ex vivo?
Yeah, so it was a myoblast line. So we tested both in healthy myoblast line as well as in a myoblast line that was deficient in GNE. So
a human one. Which is my bad.
Yes.
There you go. Some of those cell lines that were from the GNE bank, I would imagine.
Yes, so we also might do that in the future, but for now, just a myoblast line, human myoblast line.
Oh, okay. So another human myoblast line was modified.
Yes.
Great. Excellent. Let's see. Another thank you for your talk. Great. And then we already talked a little bit about GNE2 and GNE1 expression levels in muscle. Is there any evidence that expression of the aberrant gene is what causes damage to the cells as opposed to a lack of expression of normal GNE? I guess sort of is it my interpretation of that would be is it the mutant form doing something different than what you would expect the wild type healthy version of that to do? Is it causing damage actively as opposed to just the absence of it?
So as far as I'm aware now, and other labs have looked into studying that, and we see that in the patients, it is the decrease of enzymatic activity that a lot of the mutations show. So we don't expect that there'll be any negative effects of continuing to have the aberrant or mutated form of GNE in addition to the normal GNE. But that is also something that when we look to do the efficacy study in a disease mouse model, we can look to answer that question.
Yeah, certainly. I'd say it's still not totally clear exactly what the pathogenic mechanisms involved in GNEM are. So, you could envision there certainly some effects that could be related to these specific mutations that cause GNE myopathy, causing some other effect taking place. However, I think we'll have a lot more knowledge about that based on some of the studies that are currently being funded by the NDF, developing tissue-specific knockout models in mice, and I think that will help to resolve that exact question. One of the reasons that the NDF is sponsoring those studies as well as to produce models for clinical studies.
Can I just add? Yeah. So currently the prevailing model is that it's a loss of function of the GNE gene. The mutations cause the GNE protein to function less efficiently rather than the mutation causing a gain of function behavior and sort of and conferring a function that it's not supposed to do. And that's the current theory, because if mutant GNE were to confer a gain of function, then I think the patients, individuals who are carriers, meaning that they have one healthy copy and one mutant copy, would show a phenotype, but they don't. carriers of a single GNE mutation are typically healthy. Does that make sense? I hope that does.
Yeah, well, yes, but there's certainly plenty of examples where there's hypomorphic mutations where expression level is much more dependent, and you get imprecise penetrance of it. Yeah, but I mean, not to get too far off the reservation on molecular genetics in this sort of format, but no, I mean, I think you're exactly right. I mean, that is certainly the prevailing hypothesis, but I think that, at least in my opinion, we don't fully understand everything that GNE might do outside of the actual sialic acid synthesis pathway. And I'm sure that most proteins in the body do more than one thing. And I think we're still at early stage of understanding other potential functions of GNE. Another question, is there any, oh, I'm sorry, here we go. No, that's thanks for more of the information. That's great. Why is AAV the best option to use for GNEM gene therapy versus other possible viral carriers?
It's not just for GNE. It's currently known as the most efficient gene delivery vehicle for the neuromuscular field. Actually, not even neuromuscular field, for gene therapy, for the entire gene therapy world. There are certainly other non-viral vectors that are being developed, such as lipid nanoparticles and just antibody-based technologies, but those are not very efficient when you compare them to AAV gene delivery side by side. And moreover, they penetrate muscles less efficiently than AAV. And also, yeah, it's just, I feel like in terms of AAV, it's a virus that nature has sort of evolved over millions of years to be really effective at infecting cells. And we're just sort of hijacking that mechanism to make them deliver the gene that we want. And it seems that we can't, it's hard to find a replacement or sort of like a mechanism that works just as well, that's man-made. So does that sort of make sense? Yeah, there are other viruses are also being used, like lentivirus, but they each have their pros and cons. So for example, lentiviruses tend to integrate into the genome, meaning that's a potentially risky side effect in that it could insert, the entire vector could insert into a gene that may then cause that gene to be inactivated. A gene that's normally functional in human may be inactivated if an lentivirus were to insert into that region in the genome. So that's a problem. In terms of AAV, it is definitely the immune response that's currently the hot topic in the issue, and something that the field is trying to deal with, and trying to figure out the best cocktail of immunosuppression drugs in order to allow individuals, well, in order to manage the immune response in the first a few weeks post-therapy.
Definitely. Yeah, I mean, there is a reason that that multiple fields have coalesced around AAV as sort of the vanguard delivery mechanism for what is going to be for these sort of gene therapy trials. While there's a lot more technologies that are in development, it's at this point, the general consensus is it's the best vehicle available. All right, let me see. Another question here. Could induced pluripotent stem cells generated from skin cells of a potential recipient differentiated muscle cells be used as an ex vivo model to test the safety and therapy before introducing the virus into the patient proper? What would be possible, realistic, achieved in a reasonable time frame from this approach?
Yeah, I think that's.
I think Catherine was.
Go ahead, Catherine.
Yes, it's actually a project that the NDF has also funded that I've been working on extensively for the past few years, is to generate kind of a biobank of patient cell lines that were from skin biopsies. And so we actually don't need to differentiate them into pluripotent stem cells. We can actually go right from the fibroblasts from the skin cells into a more muscle-like cell. So yeah, I've been working on that for quite a while to get a biobank of a bunch of these different cell lines that not just us in our lab for the purposes of AAV testing, but also many other labs that want to study GNE myopathy to learn more about the disease or test therapies can use as a model way in culture rather than moving to a mouse model.
Great, and also we've shared our skin cells that we've collected from the patients with other labs internationally, and including Marianne Huizing from NIH, who have converted them into iPSC cells. And you're right, of course, you can convert them into myogenic cells, but I believe the question was in light of testing, was that the immune response? Was that right, Noah? I can't remember.
Well, not so much. Mostly, I think it was in the context of the immune response, whether that would be another way to test the impact.
It's hard because the immune response is something that's systemic. And it having like a cell model is very different to having like a systemic immune response.
Mm-hmm. Yeah, I think the cell models could potentially be used to test sort of efficacy if they could restore a phenotype, but it would be hard to have them test the safety in regards to an immune response at a particular dose.
Yes. I would agree with that, but I mean these are all model, all of these are model organisms that are used to test these sort of approaches. It's good to have multiple models to be able to have more confidence in any sort of product that you're going to move forward into the clinics. So that's an important element of what's taken place in some, as Katherine was mentioning, some of the NDF sponsored research has really helped to develop more of these models. That appears to be all the questions that are present in the box, so that is addressed well. There's one other question. It's more of a housekeeping question, so I'll answer that now. But if there's any more questions on the presentation, please let me know. The question that's present now is, how do we access the first talk? You can access the previous part 1 of gene therapy update. Either on the NDF website, we'll have the directions for that, or also that's within the channel for the NDF in YouTube, so you can see it there directly as well. So I'd encourage you to go back if you have not seen that first part. Also very useful and tells you a lot about the different sorts of approaches, the multiple shots on goal, as it were, that the NDF is helping to work with to try to find an effective therapeutic for GNEM. Any additional questions? If not, let's thank our speaker today for an excellent presentation. Greatly appreciated. Thank you for taking the time, and we appreciate that very much. Also in the chat box, you'll see the link for on the NDF website to be able to access that. With that being said, I think we will wrap up for today, seeing no more questions. So again, we greatly appreciate, Katherine, you taking the time to give the talk today. That's an excellent talk, and obviously a very robust discussion afterwards. So obviously that shows some of the interest here. And I'm sure there will be a great deal of interest as well for our upcoming speaker on December 9th, Dr. Paul Martin from Nationwide Children's Hospital, where he'll talk about development of a potency assay and disease models for GNE myopathy. So talk about some additional ones of those disease models we just alluded to and the importance for these sort of therapeutic development programs. So thank you all for today, for being here today, and for all the questions. And please enjoy the rest of your day, and see you next month for another portion of the GNE Symposium speaker series.